So you're good? All right, I'm gonna start getting people together. What's up, Amanda?
Hey, how are you? Hi. Okay.
Hey.
Oh, nice. Yes. I forget your name. Oh yeah, from Europe. Yeah. Okay. Yeah, you too. Yeah, how are you doing? This is like a little reunion space. All different worlds colliding.
No.
Yes. At least on me.
Is Marius here as well? Which one is Marius? Because I've seen him
Well, I haven't seen him.
twice.
Yeah, I mean, he's supposed to come. I don't know.
He's not here yet. Because I'm sure I have to, because he's a very good friend of mine.
Cool.
I need to say hi to him.
You look good.
Are you okay?
I'm okay. I'm just like really sort of discombobulated.
Why?
No reason. I just had sort of like, sometimes your brain just isn't working yet. It's like, because it's like the end of Friday. And I'm like, oh, I have to go talk. And I'm starving. And like, just. Yeah, I'm doing it. I'm actually the scientist in residence, so they have this new position there. So it's like a half-time position, which is, yeah, it's fun. I'm setting up a biolab and a class. Yeah, so it's very new. Yeah. Oh, do you have to leave like right now? Are you just visiting from?
Yeah, I'm on like I'm based in Newcastle and the culture lab, but I was
Hey.
Hello.
doing some stuff in Canada.
Yeah.
Okay, guys.
I
So
should get my computer out.
everyone grab a spot. We're gonna do 15 minutes of lightning talks, or 20 minutes, and then take a break and do some more in a little while. My name's Matt. This is Chris. And together we organized Future Lab Camp, New York City, and we could have done it without the help of Gen Space. But I'm gonna let Chris talk a little bit about the floor. I'm gonna give a little talk later too about the idea behind Future Lab Camp. But suffice to say, to start with, there's a theme. The theme is exploring biological prototyping in whatever ways might be germane. So maybe that means building actual devices. Maybe that means exploring the idea by fictionalizing the future. Maybe that just means trying to improve biological protocols that already exist. So lots of ways to try and approach that theme. And you all are gathered here because you have different levels of expertise with which to, by combining with each other, explore that theme. And so I have a fervent hope that small teams will form today, and then over the weekend we'll explore it together. So that's the basic idea. And I'm going to let Chris explain a little bit about the space here and about our first speaker.
So you at the Metropolitan Exchange, actually a former bank building. Where's Al? Is Al here? He's, you've probably seen Al, the man with the white beard. For those guys who attend the workshop, actually Al is the man who can make it happen. So on the second floor is going to be full of materials, acrylics, plywood, MDF, and a lot of like old devices and things. We have a workshop in the basement. So I'm part of this floor, and I want to say thank you for everyone who's here on the floor for the chance to make a workshop like that happen here, because we haven't done that so far. And while I rent that space right here, and I cleared out the desk and to make like more like a format with actually presentations. But so we're gonna put later on. We're gonna put up like desk spaces here, and also like for those people who are coming from not from New York, who are coming from Boston and other places, you're very welcome. I will be here all weekend, and I'll put up a tent here, and you're very welcome to join us and like And so we material. We all have a little bit of.
Yeah. So we've got there's some magical stuff downstairs. If you can talk to the man in the white here, maybe he'll let you use it. I brought some electronics, so like some Arduinos and webcams if you have. And I ordered some micros that look interesting with a microscope. So we've got some basic materials, but we also have little micro grants for all of the workshop participants. It's about $100, sponsored by our friends at Autodesk Research. So they've sponsored us, so we can give you guys $100 to go out into the neighborhood to get makeshift supplies to explore your project.
And there's actually on the blog on the futurelabcamp website, I posted a link of like the closest best hardware stores and like where to get. You can look it up. There's like the sort of like tourist guide for hackers.
Yeah. So we brought you guys together. We've got the venue. We've got a little seed funding for you guys, and we've got these talks now to help us start. So we're going to do the talks, and then after that, you're free to stick around and socialize, and then maybe we'll go out, something like that. And as Chris said, we can camp here.
Yeah. So at the workshop, we're going to start actually working on projects on Saturday, tomorrow morning. It's going to be a second session of like talks, like with people that attend the workshop, and like well, this is the first one, the opening, and the second series, and we'll have. So what we're hoping is that we'll maybe have like 4 or 5 teams, like find each other, like making a project table in the building, basically a project by the end of Sunday, and then we will do. It's going to be competitive, so we're going to have a competition. So and there will be prizes, and what we're hoping is that actually like it's not just going to be lightning talks and conferences and socializing, but also that we produce a project that can be a device, an object, a protocol, or like a video, or a website, or like it's very open. We're going to help you actually.
Suffice to say that we're going to do our best to document and record the output of the workshop so that whatever it is you guys decide to work on in your teams, they get continued in the future, either as collaborations between you and your teammates, or someone else sees it as inspired and builds on whatever it is.
And we are intending to produce actually a newspaper that's going to be like sort of a product at the end of the camp.
Right. Some of the people who came are interested in actually, as their project here, making a newspaper about the event. So that may happen, maybe it won't, maybe those people will be poached by other teams. You can never tell. Is there anything else to say? I think the only other thing I want to say is a big thank you to everybody on this floor, and especially Jen Space there's Ellen back there. She's going to talk a little later tonight about the space. Just about letting us come in and host the event. So thanks a lot to you guys for making it possible. So anything else you want to add for that? Welcome. Great. Okay, so clap for Jonah. You go up there.
Yeah.
Okay.
Hi everybody, I'm a researcher, artist, writer, PhD at Trinity College in Denver. Today I'm going to talk about some of my work on deconstructing networks. These are projects I've developed over the last few years and critically at how to interact with networks. I want to start with a quote that I think is kind of interesting in this area. It's by Tom Sandage, who wrote the Internet. It says, time traveling before arriving at the 20th century, it would surely find space-like and routine concept of air travel far more impressive than the Very close what was talking about the telegraphs and what led us to the telegraph office on the right of this phenomenon, bluejacking, where you're walking by a shop and you get a So my work kind of sits between the middle of these two areas. On the left, you have this telegraph office where it takes infrastructure, specified location, and network-dependent connectivity. So you actually have the key of this location to connect. The right side, you have mobile infrastructure. Location doesn't matter anymore, and the devices default to connectivity, so you can actually use this worldwide device to device. So I'm going to talk about projects in mind that look at this area of suburban network context. consequences aversion, we got consequences to network interaction, displays, represented data in physical space, wireless control, and wireless access in that space, and also at the end we'll talk a little bit about recycling, how we can reuse technology. So start off the network consequences. This is a project that I did called Urban Infrastructure, which is this large kind of jackhammer the size of buildings. And every time the website is issued, the jackhammer up like it's destroying. So it's physical. And it presents how website relates to efficiencies. It represents how people in those two spaces can interact. That's something that doesn't really happen all the time. The website might get more attention than the physical space and vice versa. And it has consequences, something like using an online space. There's a video, but it's probably a little bit too long. It's only like
Go for it.
but basically, it's just consists of this large drill, and then every time the site gets hit, that's the drill. And I installed it now with -50, and this is how it sits. Another kind of consequence you get when you visit your site. This is my next version, which I made. In New York, I'll have a chance. Everyone's a strong version. Looking at network subversion, this is an early project we called Complicated Web. I built this article hand crank. We actually enter the website and have to download by hand. So it cranks kind of bits into the browser on the web. Writes how much cranking will do. So it's looking at how it can materialize things like bandwidth, which are invisible. Also looking at physical interaction and whether automated, everything's automated, and much more. It also looks at a 3,000-year-old technology, use of bandwidth and internet today. So how do you kind of combine those two? It's really loud and obnoxious. Moving on from there, I'm interested in kind of representing data in new spaces, and this is a project that we see. And it's basically a data sniffing software that commands a miniature police car to drive around the platform. And every time data on the network is sniffed that has any terrorism or terrorist activity, it sends the cars into the immediate pattern. So the idea is to kind of reappropriate data in a physical way and find the way that we kind of think of data visualization as something off the screen. There's been a lot of ways to run stuff. Mine was kind of going more for the reading of the data in this space. I got strengthened the relationships. I only got time to do this. There's a video that you guys can watch. Looking at wireless control, this is a little bit less of an issue now because people have internet on their phones now. Like there's Wi-Fi users that can control the car. All the Wi-Fi signals are challenging each other for jurisdiction in the park. And so I built this project on Wi-Fi Pod because this as a jammer connected to a laptop. So that when you walk into one of the Wi-Fi hotspots, you can jam everyone out on yourself. So it's kind of glad you can control a hotspot on their own. Because a lot of the providers at the time were edging other nodes out because they wanted secret on this. And this shows you kind of how you can allow the individual to do it on themselves. It's kind of being able to sphere as a way of kind of challenging Wi-Fi in this space. And the way it works is you have Wi-Fi users, and then with the polygon, only your packets go through, and everyone else is blocked. So it's kind of just to show that that control exists from somebody outside the provider. Finally, I'll talk a little bit. This is some workshops that I do around the world. Backyard challenges. Basically, what we do is kind of hacking workshops. We take junk from places we visit. And since 2003, we've held them 46 times in 1455 continents. And basically, what we do is we kind of go to the city, find junk, bring it back to the state, work on it, and then have a performance of all of the junk to see what made, projects that people have made out of the junk. And it's really sort of about interface more than it's about exploring interface out of recycled materials, using recycled materials to create new objects. This is a video to watch out. Physics rebuild class, just to show you one example. But we do it where basically they're building controllers and interactive objects out of this stuff. Here's a example. So this is a helmet that some guy made that actually plays tones. This was a drum machine someone made out of a printer. This is a headbutt, makes some sound. Headbutt helmets. This is a drum that this guy made out of bubbles in Korea when we were over there. And this was a robot that runs over an old synthesizer and makes tones based on how it's moving. So it's all kind of about looking at, once again, sort of recycled materials and challenging that use and kind of creating new interfaces from that. And we did some public performances, such as this one in LA and in Montreal, and so did that. So there's a whole website on that. Thank you guys. I'm on.
No, that's awesome. I hope we can build some cool, maybe some lab equipment. Yeah, in a similar way to the junkyards.
Yeah.
The instruments, those are great.
A lot more. It's probably the same, similar type of aesthetic as your.
Cool.
Oh, you want to use some computer? Yeah, you can. Oh, we are doing freestyle now.
Freestyle.
So, is there someone who needs to leave early someone from the presenters? So, someone in a hurry who wants to go now? Like Jonah had to do it right now because he has to leave. No one. So
Okay.
we can like
Go back to the original order.
yeah. So wait a second, we have to restore the projector.
So I guess I'm first, actually. So let me load up my presentation.
Okay.
And then,
You're gonna do it on yours.
yeah.
So then I switch the thing. Alright.
Cool. Oh, but Chris, would you mind advancing mine, or maybe I can get an audience volunteer. Yeah, I'll get someone.
Yeah.
Oh yeah, sure. I'd love to. I just made it. There we go. So this is based on a little talk I gave about 6 months ago, and the basic thing I want to remark on is just some trends that I've noticed. And I want to think about, thanks, modularity and prototyping. And also I want to fix the projector. Actually, it doesn't. Oh well. Thanks. Do you think it works? To like sync up. So I guess what I've noticed is, does anybody know what an Arduino is? You know what one is. Who else? Some people? Raise your hands if you know.
Hi.
About half of you. Anybody know what a MakerBot is?
Oh yeah, sure.
About half. Anybody know what a personal computer is? Everybody has a computer, right? So what I want to talk about, can you be my volunteer? Sure. Is, oh, that's why it's not previews. There we go. Yeah. So you can just press the two arrow buttons. Oh, good, it does work. Okay, cool. So what I want to talk about is what's the nature of those tools, and what might be sort of a way to translate that into molecular biology as it stands today, or synthetic biology. So let me time myself too, so that's only 5 minutes long.
4 minutes, 26 seconds.
Thanks. You've already started. Okay. So basically, to me, one way to describe an engineering cycle is right here. So you have an idea, you come up with a design that implements that rough idea or the thing you imagine. Then you maybe, if you are an engineer, spend a little bit of time actually figuring out if your design will work in the real world. So you do as much work like without building anything as you can, or at least as much as is economical. Then you build it and you see if it does the thing you wanted it to do. Like the motor spins at just the right amount without blowing up. Good. Or maybe it does blow up, so you need to go back to the drawing board and adjust your design a little bit. And in this way, you end up cycling around this cycle. So you do this over and over and over again. And what I would like to say, or describe as the prototyping cycle, is you can sort of take this and move it into software, move it into physical hardware, maybe even into biology. And so if you're working with software, getting around this, going from like, you've got an idea for an algorithm or a way to make a login box on your website or a way to sort a list faster. Coming up with that idea and then testing it is very fast. That can be as fast as you can type. And so building the prototype of your idea is like a 30-second operation in some circumstances, if you're working in software. And if you're working in hardware, depending on what kind of tools you have, it might be like a several hours sort of thing. Maybe you can build a prototype pretty quickly if you know what you're doing. But in molecular biology, if you want to insert a gene X in the organism Y and turn on gene X, that's more of like a week to month-long sort of effort. It takes a really long time compared to the other two industrial domains I just described. And maybe it's comparable to like, let's build a skyscraper, right? It takes a long time and you have to get it right. So it's pretty hard. So right now in synthetic biology land, and one way to describe this new field of biology, synthetic biology, is to say it's trying to apply engineering principles to biological systems. In other words, trying to make this engineering cycle work and then go faster. These are some of the efforts that are happening. So some people are working on like this sort of informatics space, trying to describe things in a modular way that computers can understand. Some people are trying to build computer-aided design tools, sort of like Eagle for circuits, or IDE for computer programmers, but for biology. So what does that look like? That's sort of in this lower quadrant. And then there are some people who are trying to make it easier to actually build your physical design. Those things are all happening, and I know a little bit about those efforts. And so if anyone wants to discuss, like, I want to make the conceptualization part of biological engineering easier, or I want to make the construction part easier, come talk to me during the workshop, and maybe we can come up with a new approach that we can even maybe prototype this weekend. So the really interesting thing, though, is that this lower part over here of the cycle is sort of where it becomes real in the real world. And so I'm going to tell you a little bit about some of the hardware that's been happening in biosyn that might sort of fit. So here is the rotor of a centrifuge that was printed with a 3D printer. I think a MakerBot, but I'm not sure. But it was, yeah, good. And so it's certainly available on Thingiverse. So here's one example of an open source tool that helps you do biology a little bit that's now available that you can print. So I think that's sort of interesting to know. And here's a microscope that a guy who's running a workshop today, actually, on biology stuff, is playing with right there. And this is, in fact, like a pretty prototypical DIY biosyn. So you've got a biologist doing stuff who's really intense, and two guys who have no idea what's going on, so they're laughing about it. And then you've got a reporter who's like trying to make a story. So I like that one a lot. So it's pretty cool, though. This microscope design was sort of championed by that guy Mark in the green shirt. You basically take a webcam and vertical lens, and now you have a magnification factor of about 100 or so. So you can see some small things like big cells, algae, maybe even some microbes. So I brought some webcams that we can take apart and fool with, and I brought some microbes, algae and protozoa that look cool. So if anyone wants to play around with like attaching the movement of algae or microbes to PD to make sounds, or whatever. We could do that with some in this case, Mark printed what he calls a motility sorter. So cells start in the circular part here, and then if they're very active and they move a lot, they move into the square area. It's sort of like a two-dimensional minnow trap. They can't go backwards very well. And then you can watch it happening live with the camera. That's a PS3 camera, a little more expensive. I bought $5. This is what it looks like though on a screen, so it's pretty cool. You can build a microscope very cheaply. Another friend of mine's been working on these microbial batteries. So you put carbon electrodes on the top and bottom of the soil column. You put water in there, you let it sit for a while, and now you can get microvolts out of it. So tiny amounts of current, enough to blink a light, move bits. It's still pretty cool. Like you can bury one of these. It doesn't need sunlight, it doesn't need anything, it just generates current very regularly, very wide. NFC, microbial fuel cells. So those are kind of neat too. But let me get back to, so the rest of the stuff are other examples of open hardware projects. And I won't go into all of them, but here's part of the inspiration. The Arduino, some of you know what it is. It's a very nicely designed interface that's not very expensive for programming and playing with microcontrollers. Microcontrollers are cheap for computers. And so in a way, this is an enabling technology for people who don't know how to make things, how to program things, get to program things. And some of the key features of the Arduino are that it's low cost, it comes on a board that's easy to interface with and to understand and to hold. And it comes with, when you buy one, you get hundreds of example files that show you how to use it. And so this is a thing that was not designed by engineers. It was designed by interaction designers who wanted to get other interaction designers to play with microelectronics. And that to me is very important because the engineers look at this, most electrical engineers I've talked to, it's kind of scoff and they say $30 just for that mega chip? Like, that's ridiculous, that's so underpowered, it's inefficient, this is not the way that I would design it. But that's their sort of professional perspective. And what they would say is they would say, buy the chip and program it yourself. It's only $4 instead of $32. But if you look at this sort of how interested is the world in microcontrollers, if you do a search for one of the most popular microcontrollers ever, the 8051, and just the word microcontroller and the word Arduino, you can sort of see that the first two cases are coupled tightly over time. And then bam, right around 2006, the Arduino sort of was born out of an older project. And then now it's actually surpassing these other two technologies or concepts in the average internet user's mind. So I'm not saying that every electrical engineer out there should use Arduinos. I'm just saying that as a instance of a microcontroller, it's important because of the way it's been designed. It's very accessible. So here's another example, right? This is not a computer. This prints things. The founders of MakerBot is right back there. And I think this is interesting because to me it's analogous to the Arduino, but for 3D printing. So here's MakerBot, an older MakerBot, and here is sort of the cheapest pro-level version, at least according to me when I was looking for this stuff. So you can see the price difference, and if you talk to a professional person who uses rapid prototypers a lot, they would say, don't use a MakerBot, at least maybe a year ago they might have said that, don't use a MakerBot, buy this $1,600 Stratasys printer. 16,000, sorry. And as you can see, there's a similar sort of interest graph down here on the bottom, and MakerBot just totally dominating the discussion. And I think it's dominating it because it has similar properties to Arduino's. It's low cost, it's okay to mess it up or to change it around, and it comes with a community of people who provide a lot of examples on how to get started with it. So those are the properties that I think are really interesting in these tools, and in my mind, the Arduino and the MakerBot are examples of things that make it easier to prototype in different domains. So if you want to do physical computing, you use the Arduino. If you want to do 3D prototyping, maybe you use the MakerBot because it's so inexpensive. You can kind of hack it yourself. So therefore, the question is like, what is that in biology? At least according to me, I mean, biology is a very big thing. So maybe it's too general to just say in biology. But let's be more specific. So if you want to do biological engineering, specifically if you want to identify a gene that some scientist found somewhere in the world and it's in an online database, and now you want to see if it does the thing that they say it does, how quickly can you do that? And right now that's sort of a month-long project for, I don't know, $5,000, maybe $1,000, somewhere in there. So it takes a long time, it takes a lot of technical knowledge. There aren't really good examples that help you get started, and it's kind of expensive. So maybe by kind of hacking together the existing set of biological, low-cost biological tools, exists now, maybe we can start to build that thing that lets you get around the cycle faster. And to me, I think the way that we can work on that in this workshop is by working on the hardware side. So let's try and like mash some of these up. PCR machine with a robot on a MakerBot with a gel box. So you press a button and it does everything. At least it does PCR and then runs it on a gel. So I mean, that would be pretty cool. So that's kind of what I hope. And eventually, I just wanted to end by saying, if you think about a machine shop where a guy's making precision plastic components for something, you've got a guy in a room and he's moving around with like some substrate, like a block of like Teflon or something, putting it in different machines until he's implemented the shape he wants. And now think about a MakerBot, this one machine that plugs into a computer, you download the file you want, and you print it. Maybe it's not quite as precise as the guy in the shop, but it certainly was easier to do. And in a way, you can kind of encapsulate the entire process of making that object into this one machine. It's a physical object. And so when you want to put a gene into an organism, like say microbiology, you might put a growing gene into E. coli, you've got a scientist in a lab moving the gene amongst different pieces, or moving bits of DNA and microbes amongst different pieces of equipment, and trying to do basically the same thing. Like if you take off your glasses and you look, is it in a machine shop or is it in a lab? It's basically the same. So by encapsulating that into one system, the entire process becomes easier. And I'd like to see us try and do that. And just to be fair, DIY Bio, which is where some of those hardware objects came from, has not experienced that sort of amazing rise. So I'm not sure if it will ever happen, but we'll see. So thanks for listening to my talk. Okay, Chris, who's next?
Yeah, I have it right here.
Okay. Maybe we should stick to that.
You're gonna stick to the list.
Yes. It's gonna be very German.
So,
That means Marius.
ready to go?
Oh, okay. So maybe then Marius and then you.
Okay, great. You can go first if you want.
Yeah, maybe you.
All right, yeah, actually,
Yes. So let me check. Wait, I'll bring up the presentation. Let's see. Is this the one? Yes. It's coming, it's coming.
But yeah, it's going to take over your keyboard for a second. Just so you know.
What?
Alright. So while Chris is doing that, I have an idea for our sort of. Oh, you're done? Okay, good.
Yes, very cool.
I'll continue that sentence after the next presentation.
actually, it's funny because if you follow your analogy to the end, what you need is an immediate DNA sequencer. Because that is all. It's not sequencer, it's a synthesizer. And it synthesizes whatever you want, just like a MakerBot, and then inject it into the organism in your cells.
Can you just say next, then I'm going next slide?
I've
Or
got,
you?
I've got the power.
You can control me.
I believe so.
Okay.
Yes, this is
Does it work though before you get to the.
Okay. Then just say next.
All right.
It's blinking.
Okay, I'm Ellen. I'm at the moment president of GenSpace. GenSpace is a community lab space, and what that means is we accept members for a nominal fee. You get the use of the facilities, and we share some of the expenses for the consumables. If you have an exotic project, you buy your own stuff and bring it in. But pretty much it's worked out so far. We've been open since December. And as far as we know, the first one with this particular model that's up and running, although, I mean, there may be labs somewhere in the world that we don't know about. And certainly a lot of people in the United States have been, way before us, have been giving, they gave us the idea to do it. Actually, I'm wearing a t-shirt from BioCurious, which is a lab out in California that's starting up now too. Okay, next. All right, well, I don't know if anybody has seen any of these headlines. This was kind of apropos of what Matt was saying, how there's always a reporter looking over our shoulders. DIY Bio was really big news, especially a couple of years ago, and most of it was rise of synthetic biology, which is sort of the intersection of engineering and biology, the way Matt described it so elegantly, and the way Oliver's going to talk later in this event about it more specifically. And the fact that the cost of these things, I actually have over there 39 cents a base per, I think it's down to 26 now. It keeps getting cheaper and cheaper to do genetic engineering and synthetic biology. So it's the interest combined with the means. Next. So this is Gen Space. That was at our opening party in December. 1, 2, 3, 4, 5 of us are here right now. And you can talk to any one of us in depth about it if you're interested in the lab or being a part of the lab or just what we do. Next. We got together because we wanted to do cool things like turn bacteria green. Next. We started out in Dan's living room with a dark slope. There is the ever-present reporter in the background. People who are non-scientists laughing. Exactly the same scenario. Next. That was one of our first DIY efforts. It's really pathetic. It's a gel box. Next. That's Marie having fun taking pictures of us doing science. Next. This is what kind of the public's view of what we were doing was. So we realized if we were going to do this any further, particularly with all this bad press coming out, that we would have to do something, we would have to reach out to the community, we would have to build a dedicated space maybe. We started making friends. Next.
Oh, I want to take a picture of this.
Okay. So any friend of Mac is a friend of ours. You may not recognize this gentleman, but he is the head of the Weapons of Mass Destruction Department of the FBI. Next. So we ended up at conferences with those people, and we did a lot of other sort of proactive stuff. At any rate, when we went to do a dedicated space, we actually went to New York Resistor, which Marie has been a part of for quite some time. Were you one of the founders?
It's my fault.
Oh, okay, it's your fault. Good. But I remember early on we came to you and you said, do you have any guidelines for us? Do you have any advice? And we really used a lot of what you told us. Probably the only thing we didn't do is incorporate some floor prop at the end of it. We're still wondering whether that's a mistake.
We want to go for the map. We are good.
Yeah. At any rate, so yeah, actually one of our first attempt to sort of get more membership was in the New York Resistor. We used to be there, so Jack found the Max, which is an awesome space. So, and we want to go back actually and do some more stuff with you guys in the settlement. Next. So this is this is us fixing up that lab space back there. You can just go through these slides. Next. Yes, this is all of our. And okay, stop. So that's what it looks like now, pretty much. That was one of our classes. We run crash course classes here now, almost all the time every weekend, and we get a very wide variety of people interested. What I thought was really cool was two people interested in the biology of yeast, because one was a whiskey distiller and the other was a winemaker who showed up at our first class, and they wanted to know more about genetics. Next. This is just, I guess, keep going. I think I just want to put more pictures of the space. Yeah, that's pretty much the end. We're happy to have this gathering here. We think it's really cool. We'll be around on and off, some of us. I think Son's going to be here for the whole event. Are you, Russ, going to be here for the whole event?
Here for most of the time.
Yeah, and Oliver and I will be here tomorrow as well. So if you want to talk to us or collaborate with us, we do anything. We have two labs back there. The big glass one will be closed off because we have precious samples and things in there. And the members just felt more comfortable closing it off. But there's a second lab there that's open that you can use, and there's equipment in there. And Matt brought some amazing equipment with him. So thank you very much, Matt, for hauling all that shit down here. Okay, I'm done. Thank you.
Thanks. So Adam? Is it Adam?
Adam, yeah.
Cool. So in the meantime, I was thinking maybe we could fill the time while people are setting up by just doing short introductions. Let's go in a clockwise direction from where I'm sitting. So, oh no, from where the screen is. So clockwise from the screen. That would be you. Why don't you give like 3 words that are in your tag cloud? The 3 words. What are the 3 words?
I'm thinking from Switzerland.
Swiss visitor. Okay. How about you?
I'm Corey. I'm actually one of the manufacturers. I deal with materials fabrication and I guess bioanalysis, sustainability, etc.
Okay.
I'm Beatrice. I was speaking later, and I'm very
Okay. So now why don't you start? No, we're gonna do it every time.
Do I need the little?
Does it work yet? I think it might be working.
That's at my. I don't know how does it work. If I can connect this to Bluetooth.
No, it's already connected, dude.
To my what?
Yeah, I plugged it in. It's right here.
Oh, that one.
Yeah.
All right. Let's see if that works. Can you try it? Yeah. Something happened. Yeah, which is good. Let's do it. Cool.
Yeah.
Awesome.
All right. So we just came to the building. We would see and our team from the city in the building 9 months ago. And so this is one of our first event here in the space. So very good to be here. So we've long been interested at Seed about how to use design to communicate and help people understand really complex language. It could be biological, ecological, environmental, physics, whatever the case may be. And recently, really over the last couple of years, we've gotten really interested in how data visualization can help us better interact with big scientific issues, big scientific concepts, and start creating more connection between scientists and designers, with data visualization being really the interface that allows for these types of communications really nice. And so a few months ago, we launched this new project called Visualizing.org, which is an open community, an open platform to let you explore and share data visualizations with a large focus on complex issues that have some sort of scientific orientation to them. So there's quite a bit of stuff on here. There's several hundred visualizations that have been uploaded over the last few months, all under a Creative Commons attribution share alike non-commercial license. So you can go on visualizing now, upload your work, share your work, and find visualizations of the several hundreds that have been uploaded on global health, environmental issues, sustainability, biological issues, all sorts of things. And what we've started to do is try to get these visualizations into the mainstream discussions as things are happening in the world. So as issues like Japan take shape, visualizing start to integrate visualizations into the conversation. So lots of really interesting things happening just with sort of the news cycle changing over the last few months through visualizing. The other thing you can do, and this may be sort of an interesting resource if you're sort of thinking about the possibilities of data visualization as an interface as a project, is we've been working with lots of major research organizations and NGOs around the world to pull together data so you can find a ton of data on ecological issues, biological issues, environmental, and everything else as well, and grab all that data, all of it's free and open, and sort of use that to create content. So it's a resource, it's free, it's open, and invite you to use it. So what I want to show you is a couple of kind of really interesting projects that have been uploaded on the site just in the last month or so, and kind of some themes that are starting to emerge. And these are all trying to just focus these on stuff that's happening in a biological, ecological, sort of environmental context. So some interesting projects now, they're starting to take biological data and put it in the hands of designers. So there's always been a school of thought, of work, taking biological data and obviously information design, bioinformatics around the biological data. But now this data is moving into the hands of sort of amateur designers who are looking at issues like biodiversity, creating really interesting heat maps to look at biodiversity around the world using open data sets, looking at a personal project, for example, on synesthesia, and kind of looking to map how an individual might look at and see things from a personal perspective. Projects are starting to look at relationships between diseases, relationships between genes in a really interesting way. Trying to map biological data, but again, from the sort of design community standpoint. Another really interesting kind of avenue is looking for interconnectedness, and this is an area that we're really interested in. We're doing a lot of work on. We're trying to map things like someone's ecological footprint or a country's ecological footprint with biodiversity and the consequences of biodiversity. We're looking at what are sort of the economic benefits of stopping deforestation. So really cool projects over here. This is a project connecting public health and green space. So if you start mapping green space in a particular environment, you map that data to the public health in that particular country or city. Really, and some interesting metaphors looking at green space and then mapping that to public health. You heard a little bit about physical data at the beginning. So some really awesome projects here. This is one that just came about. We just did a World Water Day challenge. So this is pouring an equivalent amount of water on each country based on its increase in urban domestic water use over the last period of time, and then seeing what the resulting consequences are around the world. Using origami to look at what are the factors that keep the planet in sync, kind of looking at overall planetary boundaries, sustainable boundaries for habitability, and what are the sort of, these are data representations. Looking at fresh water withdrawal. Interesting kind of folding projects based on a person's CO2 emissions and how to reduce them. Really, really, really cool stuff. This is really taking it down to an even more personal level. So lots of projects that are trying to take some, sort of from the personal standpoint, these are my emissions. This is sort of what I'm doing on a daily basis. These are products and things I'm consuming that actually sort of match for me personally. This is a really cool project called Habits. So it's designed to pull data from a community of individuals and then reflect it in sort of these morphed configurations. With each body part representing an aspect of the sort of daily CO2 emissions. So feet, for example, representing transportation. So depending on modes of transportation, every part of the body and what swells. Really cool budget. This is sort of really simple kind of day in the life stuff. A lot of designers, a lot of people just doing that. We did a hackathon at IBM a few months ago, and a lot of projects came out looking like that. Projects that were just very personal. And these are really interesting kind of dashboards to look at everything you do in a day and looking at these sort of environmental. Kind of a final project, something that we're really, really, really interested in. We're getting involved in some of this. So this is a group called Amano, working quite a bit in Central America, doing some work in Honduras, for example. The idea is how can you bring a crowd together, bring a local community together to start mapping their environment. And either mapping terrain, mapping sort of environments that have not yet been fully mapped. We're actually taking things like biodiversity, looking for specific species, looking for particular data sets that they found that by engaging the community, they're creating sort of a map together that's very physical, very analog, and then it gets digitized, it ultimately gets shared, it ultimately gets put on the web. But it's actually starting to have an impact on local policy because the community is kind of producing the data, sharing the data, and kind of collectively mapping it. It's starting to actually impact the way local government is enacting policy around these kinds of things in its community. So a really interesting kind of new notion of how a community will come together, contribute its own data, and get the kind of systems picture that we often don't get if we're just contributing a small piece of the problem. So it's immediate, it's responsive, and it's actually having some really cool impact. So that's it. I just want to share with you some projects. You can go up on visualizing. Hopefully it's a sort of source of inspiration if you're interested in kind of data-based projects. Certainly upload under a CC license that we find data and let that help you. Thanks, guys.
So that makes Rich you next in our introduction line.
Hi. I'm Simon from Pittsburgh, and from the Center for Post-Natural History.
And what about you? You're Ellen.
Oh, I'm Ellen.
I guess everyone knows you.
Molecular biology.
Okay, let's branch off from here. What about you in the white shirt?
I'm Una. I teach in Northeast E. I was in the lab, and I'm interested in product design and function design.
And tap someone in your little cluster there. Next.
So I'm Russell. I'm a co-founder of GenSpace. I used to work at NYU, graduated there last year. Now I work at Cornell Med, defending their iGen program, which is an undergraduate competition for synthetic biologies.
Cool. How about you?
Okay, I'm Caitlin. I'm just a part of pieces and have interest, but this is not actually my field. But I'm just very keen to
Cool. Yeah. How about you?
I'm Toby. I'm at ITP. I'm at ITP right now. I'm interested in biology, but probably my expertise would be in programming and physical design. I'd be interested in doing any sort of crazy mashup programming stuff. Is usually what I end up doing at these like hackathon things.
Man with experience. Okay, so are we ready to do the?
Yeah, who wants to go next? Wait, who's like?
Oh, I thought you said.
Marius. Marius is next. So you want to use your computer, right? Oh, okay. What kind of plugin do you need for this video? Just
Old school, man.
oh yeah, okay. We can. No, that PC. Awesome.
That's all you need.
All right.
Okay, so let's see.
Want me to advance your slides?
No. I'll have to operate it in here. So I actually just realized that when Chris invited me, I just sort of sent him a proposal for a project that I could show, and he said, yes, let's do that. But I'm realizing that actually that's just kind of like an artist presentation. So maybe it's more interesting if I actually do the presentation I was supposed to do at MakerBot on Wednesday about my residency for MakerBot. It's a bit more hacky.
Yeah.
So I should do that? Okay. So I'm flexible. All right, so so this presentation is called For the Love of Plastic, which, you know,
You're a plastic kind of person.
well, at least for this residency at MakerBot. So Bree invited me to work with MakerBot for their first ever artist in residence program, which I thought was a really great initiative because the MakerBot has a really great presence among hackers and DIY people, and you know, like the user group is like, I met more engineers in one night than I've met, you know, the last 3 years. So that's really interesting. But at the same time, I saw there's only 2 architects. So I think like if we could balance those numbers out and make the hacker DIY engineers meet the architects, it's something really awesome. So my personal work is I'm a visual artist. I'm a formalist. I'm not particularly hacky, but I do write code because the only way that I make these things are by writing code. So I've been interested in kind of generative articulation of form for a very long time, for about probably 20 years at this point. And in the mid-90s, I heard about 3D printers for the first time. And they sound like, you know, just sort of magical machines. These pieces were the first things that I ever did with 3D printing. These are about 4 inches in diameter. And there's something really interesting that happens in that kind of micro space. These, you know, I would love to build these things the size of houses. Kind of did recently, but for now, 3D printers are more useful for sort of micro forms. So Bree contacted me because of this work and because I've been writing libraries for processing. I wrote like an STL output library for processing a while back, and I've been very involved in the processing environment. So we talked about how could I bring this work into the world of the MakerBot. Now, you know, writing STL code is not very hard. It's a very simple format. The challenge is really how do you get into a convenient computational workflow for creating mesh geometries. I don't know how many of you here write sort of computational polygon meshes. Yeah. So then you know there's like a ton of vector math and you know a lot of sort of good thinking, and just it's not the easiest thing to do. So I decided that in the course of the MakerBot residency, I wanted to produce a bunch of models, which Bree and his bot farm has really been very helpful in. I think I sent some models down, and they, you know, and the MakerBot crew went to Las Vegas for the CES, and then they came back again, and I showed up, and they had just printed like 30 of these things. So again, you know, for the love of plastic. So these are all pretty simple in terms of the algorithm behind them, but what I was interested in was, okay, so how can you take the MakerBot, which is really known for its, you know, ability to sort of fill in for a lot of, you know, higher end 3D printers, and actually go towards a aesthetic direction. What are the aesthetic properties of the MakerBot? And they're actually, I mean, you can kind of see it here. It has this really lovely vertical resolution as you build up the pieces, with the little ABS plastic spaghetti threads. So I thought it was quite interesting to just look at that on its own. And at the same time, I was planning this solo show where for the first time I was planning to build some large structures. These are the MakerBot models, like on proper display and art gallery. And I was working with a much larger object. These are cutting tabs for plywood panels. And this was a model that I built with this library that I've now released called Model Builder. And Model Builder is just kind of a set of convenience methods for articulating meshes out of sort of simple metaphors, like, okay, you take a vertex list, and you just take, each vertex list is a face, or like an edge, and then you have another vertex list, and together they make up an actual strip of meshes and stuff like that. So, so for me, it was interesting to at the same time be working on with the MakerBot, but with exactly the same tools, be articulating this shape, which this was the first model that I ever did of this thing. This was literally two hours before we started, like just CNC milling like a bunch of giant plywood plates, and these are the final pieces. So to me, what's interesting is that how the same workflow can go, from processing into an STL file and then outputting it via MakerBot, which is really quite easy to do now. But at the same time, you can plan it out, and you can be the happy artist darting around the giant plywood bookshelves. So that was quite fun to do. So the Model Builder library is now public in very much the MakerBot sort of spirit. I've open sourced these models, and we did an exhibition, and people were like, "So, how much is this thing? And how much could I buy this for?" And I was kind of at a loss, and I've kind of made a sort of my mind about how much I would charge for them now. But it's more just like, well, you can print it out on your own, in which case it'll only cost you plastic, or I can print it out and I can sign it, and it'll cost you a little more. So that's the sort of the happy intersection between being a professional artist and trying to pay the rent, and being interested in open source at the same time. And then I did these awesome lovely things with Carson Schmidt's Toxiclips library, which really doesn't have very much to do with my practice, but it's lots competitive. So, yeah, I think that's basically it.
Cool. Yeah.
All right.
What's the phone?
Oh, okay. There's this one.
Oh,
this
you
one?
don't.
Which one do you need? I have other ones too.
I've got one on my desk, but I
Oh,
need to
okay.
go get it. Do the introduction.
When I have all your presentation on
No, I might actually just present from the
So
website.
I guess this is a good time to continue the introduction circle. But now I'm
It seems like for the
at
group,
the
I should
I get to
maybe
go. So I'm Matt
show
Cowell,
more some
from
webware stuff
Boston.
and not just
I'm into
the
DIY
actual
bio
photos
stuff,
content.
and
great.
I have a laundry list of
But I
cool
need
pieces of
access to
hardware
a
that could duplicate some of the functions of much more expensive pieces of hardware in normal labs. I'm excited to
And
just
which one is connected
explore
to
some of those with a group of people this weekend.
projector?
So I'm into open source hardware and trying to disrupt the
Yeah,
system of expensive tools
I
that
don't
exist in biology.
How are you?
No, I'm just going to
Mark, I'm going to be speaking.
That's my website.
I'm an evolutionary
But I would
biologist.
love to sit.
Yeah, you can do it from biological side if you want.
Cool. Let's go down to you and then come back. So,
Hi, my name's Frank. I'm a product designer with a background in biology. Interested in ecological impacts.
cool.
Hi, I'm Nidhi. I started out as a biologist, but then I'm very warmed up to science writing, so I guess I'm your token reporter for the event. So I've been writing the past couple of months for Motion Review and technology, but I'll be doing some of the lovely things that
He's presenting for us and then you can go. It's like, okay.
Cool. And are you good? Oh, you
He's here.
olfactory stuff? Let's see.
I'm Kia Stark. I'm a fiction writer and I teach at ITP. I teach classes about relational technology and human social dynamics.
Cool. So I think we're on a roll, so let's finish it up. So
I'm great. You can blame me for many things. I make things.
you stop.
I'm Marius. I blame Free for many things.
Meta.
I'm Norwegian by origin. I'm based in New York now.
How is it?
I'm an environmental designer, urban designer, training with technologists and hackers at heart.
And it's high back here.
Hi, I'm Marie.
Cool. And Rich, you went already. And that's the white bearded man. Al, right?
Yes. And I guess I'm the gather up water.
Okay. We did it. We got through everybody. So, want to roll?
Sure. My name is James Bowden. I'm an interaction designer. My office is right over there behind the screen. I don't really know anything about biology. The last class I took, I think, was in the 9th grade. But by talking to the GenSpace folks, I've become convinced that it's kind of the next big thing. So I'm really excited to be part of this event and have a chance to tell you all a little bit about what I do. Since I only have 5 minutes, I'm just going to show you a couple graphics. There's a lot more on my website if you want to check that out. The first product I want to tell you about is called the Corporate Fallout Detector. This is something that I did back in 2005. It's an ethical shopping aid. It helps you make your decisions about shopping as you scan products for the store. And so I became excited about this because, when you go through a typical grocery store, you're kind of bombarded by these colorful products and made by a few different companies. But it's pretty hard to tell sort of what's behind these products just by looking at the boxes. So I wanted to kind of make a machine, or sort of a scientific instrument, if you will, and try to help you understand what is really behind each of these products. And I was kind of struck by the aesthetics of Geiger counters and the whole metaphor of radiation as this thing that's invisible, it's very difficult to understand, but it can be harmful. And these Geiger counters, they have this very sort of specific aesthetic that's used. I find them really beautiful. So I wanted to kind of explore how you could play with this metaphor of radiation in the process of building something that would help people shop. Okay, there's no sound. There's an infomercial about this project on my website, just feel free to check it out. Basically we built this machine which has a barcode scanner inside, and it's got a database of ethical and environmental records of about 14,000 different companies. And you scan the barcode on the product, and it makes this clicking noise just like a Geiger counter. And depending on the ethical or environmental record, the worse the company is doing, the more clicking you put it. So here's a picture of the device. And so there's a little switch here. So if you're interested in environmental effects, then you can flip the switch to dirty. Or if you want to hear about ethical things, you can just flip it to unethical. And if there's a product that you really like, but this thing is saying something bad about it, you can turn down the sensitivity so that you'll feel okay about buying it. It's this big box. It's intended to be something you kind of wear. You really this was back in 2005 before the iPhone and all of that stuff. Of course, now you can do this kind of thing on the smartphone. But I really, when building this project, I wanted to build something that would really attract the attention in the store. Where, when you would see someone scan a product and this thing would make a lot of noise, you would think, wow, what is wrong with that cereal? That's my favorite cereal. So it was intended to be more of a performative thing than something purely designed for the convenience of the person using it. And in terms of how it works, a barcode is divided into different sections, and you can take some of those numbers and there's map to the manufacturer. You can map that to the name of the company, and then there are databases online which will tell you, what company owns that company, what company owns that company, and then you can kind of collect technical data about what that company's done. For example, I think Kellogg's was owned by, lots of different companies are owned by Altria, which also owns Philip Morris, and so there are databases that will tell you, well, these companies are all owned by this UHC, this ultimate holding company, and so you can kind of map ethical, decisions that one company makes onto all of the other companies that are owned by that company. And so you can also do similar things with pollution. There's a database called scorecard.org, which will tell you for all the different facilities that, by zip code, or you can search it a bunch of different ways, how much do they pollute. And so I wrote some software to kind of collect all the barcode data, all the ethical data, all the environmental data, and kind of merge it into one big database, and then kind of distill that into basically a number for each company. And part of what this project is about is how reasonable is it to really distill all of these very complex decisions and behaviors into a number. And I was kind of trying to point out the absurdity of that, but I really think that actually you can, depending, I think on a personal basis, it's possible to make these kinds of technical decisions in that way. But really the a more personalized way to do it than just this kind of beggar calculator device. But those were kind of some of the kinds of questions that I was trying to explore with this project. And in a way, this project is kind of about measuring something that's invisible to the naked eye. And this is some more recent work here. Also along the lines of something that's one of these interactive exhibit which opened in the spring of 2010 at Chicago's museum. It's something else that's related to the idea of being able to observe things that are invisible to the naked eye. These days I run a little design firm located right over there, and what I'm working on is, this is one of the projects that I did last year. It's an interactive periodic table. I do a lot of things with different tabletop sensing technologies. So here we've got an interactive periodic table. You can place objects on the table and use them to grab elements off the periodic table, and then bring those elements together to cause chemical reactions to happen. The system has a database of about 400 different chemical reactions, and so whenever you bring things together, it checks to see if there's a reaction, and maybe it makes a suggestion, so here it's saying, hey, add some more sodium. And so we grab a cup and put one of the sodium atom and hold it into the reaction. So now we're combining that with chlorine, we get sodium chloride, and so salt pours out onto the tabletop. This is in the Museum of Science and Industry in Chicago. Thousands of people use it each day. It's really exciting to see kids use these kind of technologies. I really believe that if you use physical objects to interact with information, it fundamentally changes the way you experience it. The physical, touching things is the first way that we learn to interact with the world. And it's really just a fundamentally different experience than, say, interacting verbally or interactively. And so this is the kind of thing that kids will come to this exhibit, and the parents will have to say, okay, let's check out the other stuff in the museum. It's time to leave now. Kids really get immersed into it, and that's really exciting to see. So this whole video is something I'm very excited to check out. This is based on a tabletop sensing platform that we developed called SenseTable. It uses capacitance to track a bunch of different objects that have a 64-bit digital ID on each one. And that's pretty much it. If
You're going next.
Hey, Usman?
Usman? Did you? Oh, you got the.
I've got Usman up.
Oh, I just sent him an email.
Hey,
Yeah,
Usman.
okay, let's do that.
Okay, sweet.
Plug it in.
So we've got this guy Usman from Patchabay. Some of you know. Actually, Usman, how do you pronounce it? Oh, good. I figured it out. Okay, so
We can also connect to the boombox.
yeah, let's connect to the boombox and let's get my. Where's the projector dongle, Chris? The white one. Yeah, a little bit of light. Thanks. Just a second, Usman. Chris, where did you put the video connector cable? When we, James, where did you put the? Yeah. No, don't do that. James. Hey, Usman, I'm Mac, and here's some of the people in the venue. We're gonna get you on the big screen in just a second.
And
So that's the good.
now we give you like proper sound system.
Good. So where's the video connector?
Is that working? Yeah. I opened a beer with it.
Usman, try saying something.
Oh, it's here. There you go.
Okay. Usman, I'm going to be pretty amazed if this works because, like, this sort of thing never works. No one can hear you yet. Okay. Usman, say something. Louder, louder.
Yeah. Yeah.
Yay! I can't believe it's working. Yeah, you can see yourself.
I can see myself, now I'm getting a little bashful.
Okay, so you want to do a little intro?
Yeah, shall I kick off?
Yeah, go for it.
Well, hello everybody, I'm Usman, founder of Patchabay. We're actually having this International Internet of Things hackathon here. We're now almost 1 a.m. And I'll just show you some of the people still left over here. We've got about 20, 25 working through the night in different areas of this room. And they're doing everything from working with GPRS mode to working with a device called a Nano, which is basically an Arduino with an Ethernet shield on it, that's made very cheap. And the reason that everyone is here is around this concept of the Internet of Things, which is basically connected devices, devices connecting to each other. And what we've done is we've built a system called Patchabay, which is essentially a kind of data broker for devices to share their sensor data in real time. And what I'm going to do is I'm going to screen share so that you can, so I'll just show you very briefly the website. So can you see my screen?
Not yet. Not yet.
We can actually. But
Oh,
there's
wait,
nothing on there.
Is that his screen or is that my screen?
That's his screen.
No, that's my screen.
Oh, that's your screen. Okay.
You might have to make me bigger, like expand me or something like that.
Okay. If this was an iPad or something, a multi-touch screen, I could just go up there. I see. So no, I think you have to share it again. It hasn't come through yet.
Okay, let me stop screen sharing.
You can debug it by looking at what you're sending us.
And let me share full screen again. Let's see if that works. According to this, I am sharing my screen.
Oh, okay. Yeah, no, it is. It is. It was working. Sorry.
Can you actually see
Yeah.
the website now?
Just a second. Yeah. It's on my other monitor.
Just turn on the.
Got it.
There we go.
Okay. So
This is PatchBay. If you go to the website, and the first thing you see is this map, which basically shows you the last 600 devices around the world to have updated, probably in the last few minutes. You can click any one of these markers, and it'll show you something about that feed. You click the title, and you get to you can see something about its data streams. Here we've got something that's got humidity, temperature, brightness. There's a bunch of links which are links to its real-time data in a variety of data formats: XML, CSV, JSON, and to give you an idea of the range of different things that are connected up to PatchBay, if you click Cloud View, it shows you kind of a list of tags. And this page just goes on for miles and miles and miles. I won't actually go through it now because there are really tens of thousands of different tags. You can also browse feeds using List View. And in List View, you'll see, again, just an endless lists of all the devices and environments that are plugged into PatchBay. The point really here is to do 2 or 3 things. One is just to make it really easy to store and share data in real time. So PatchBay acts as a kind of a storage system where you push data to it, and then you can retrieve it in different formats, or you can create graphs, or you can actually send it to any one of the apps. There's now sort of dozens of apps in our apps repository, which I'm just loading up now. Different visualization tools, different dashboards and controllers, different mapping tools. The way it became very interesting recently was because in Japan, of course, with this the nuclear situation, people became very concerned about real-time radiation levels around the country, and so they ended up using PatchBay as the data broker for sharing this data. And so, is the feed gone?
No, it's good.
Still there, right?
Still my power. That's all.
Okay. And what happened was that we now have hundreds of feeds of radiation data on PatchBay, and so now you can do things like you can build this app, which is something that I built, which is essentially a way of mapping the radiation data across Japan. These are the sort of real-time values. It'll take just a few seconds to load, I suppose, especially since I'm using up the bandwidth on Skype. But the point here is that the data is crowdsourced because different people have been using different types of sensors, different Geiger counters, different dosimeters, and it was really a community initiative to focus in on the standard way to describe the feeds, the standardized units, and all these kinds of things. And this enables you to have this sort of real-time shot of what's going on in the space or in the country at much higher resolution than you might, of course, with an official data set. And it's also really about enabling people to measure their environment, to make sense of their environment, and then to share that data with others. What we've released at the hackathon here in London is it's a very early beta version of a kind of app hosting system, which is at beta.apps.patchbay.com. And here, basically, you can create apps and then share them with other people automatically. And this is where you might have a sort of script running to whatever, do a mashup, or to convert data streams from one format to another, or what have you. So I guess
Usman, how do most people connect and produce the data feeds? Are they using a special PatchBay widget, or like a physical object, or what? Is it a laptop or Arduino?
We now have tens of thousands of users, so there's really no typical way. Actually, there are a lot of people using Arduino as a kind of gateway to a bunch of sensors. There are people who have got entire sort of computers devoted to, let's say, a building management system. There's actually somebody here from New York who's got something called OpenRTMS, which is basically has an Arduino Bluetooth that connects to an Android client that pushes data to PatchBay. There are people who have sort of server-side scripts that are perhaps web scraping. Then there are a lot of devices that maybe have PatchBay in their firmware,
the current cost, for example, the energy monitor for current cost is a company that is using the API so that their devices out of the box connect to the back end. Then there are, for example, utility companies that might be pushing data just using some kind of server-side process in order to make certain data available. So there's no real typical way. It's the RESTful interface. So it's really anything that is connected to the network and can make these RESTful requests that pushes the data. And one of the things that was quite exciting about our hackathon today is that Arquessa, which is a company that does SIM cards with static IP addresses, which is unusual. GPRS modem, normally you wouldn't have a static IP address across networks. But they came and they brought their routers and modems and some SIM cards so that you can connect straight to CacheBay, actually just using a GSM modem. Yeah, there's lots of different ways.
So it sounds like there's also a New York City CacheBay hackathon too?
That's right. It's being hosted at Natalie's lab, Natalie Jernchenko, who I think is talking at your guy.
At some point. She might be stuck in her lab still. She's going to come at some point.
She was actually just pinging me on Skype as I was talking.
If you could just pass her video Skype feed to us, that'd be fine.
I was actually going to try to screen share to you with her, but okay.
Well, have you seen any biologists or scientists besides people interested in geology or radiation using PatchBay yet? Or maybe small bio or small science is really what I'm getting at. Have they been
I was actually going to say that there's increasingly marine scientists using it. So, for example, they got sensors out in the sea. To be absolutely frank, I actually don't even know what's on PatchBay anymore because we've got so many tens of thousands of data streams, and so I haven't really been able to keep it up. I'm actually doing a search right now just on biology in PatchBay, and I see there's some biological station that's coming up in Finland. I'm not sure what that is. It's worth actually just using list view and doing a search and seeing what there is, see if there's anything specific to your particular interest.
Cool. We'll check it out this weekend, and I can say for one that I had a microbial fuel cell. It's this little chunk of mud with microbes in it that generated a small current, like a tiny current. And for a little while, I was logging the temperature and the voltage of the fuel cell on the PatchBay. So I've done it once or twice, and I'm sure that there's other opportunities that we might find this weekend to syndicate the data through the surface. So it's exciting. Thanks for joining us. And does anyone have any questions for Usman about PatchBay? No? Okay then. Well, let's thank Usman. And is your hackathon ending tonight, or is it a weekend thing?
It's a 24-hour event, so we've got another 13 hours to go.
Okay. Well, if we think of anything, we'll get back to you.
Please do. I visit New York quite often, so I'd love to come and say hi at some point.
Okay, it's a plan. Thanks.
Bye, everyone.
Bye. Cool. Well, it looks like Skype virtual conference is getting better. That's exciting. So let's see, who's next? Pull up our list. Incidentally, the PatchBay hackathon people, and there's several other hackathons happening this weekend. We all sort of emailed beforehand and decided to join the PatchBay IRC channel. So if any of you guys want to find out what's happening at 3 or 4 other hackathons, Cybarcamp, PatchBay, and then Hackteria.org is running one in Columbia and one in Zurich. They're all happening this weekend with different focuses, but all sort of based on science. There may be people discussing what's up in this chat room, and you can get to the chat room just by going to the New York City page on the Future Lab Camp website, or you can join PatchBay on IRC on Freenode if you are more versed in IRC. So that's happening right now, and yeah, let's see who's next. So Beatrice, yeah. Do you have your own computer, or want to use mine? Did you put it on Chris's computer earlier?
Somebody downloaded it because I didn't finish it.
Is it?
No, it was on my server, and I know somebody downloaded on
Yeah,
some
I think
computer.
Chris seems to have walked off with his laptop. So
Let's come back to
Can I go?
yeah, you can go. So we'll do you in a second. You ready?
Yeah.
You got it on there?
Yeah.
You need the plug here?
Do I need a...
Here you go. We can just trade spots.
This might be... I need my...
Need yours? Okay. Okay, so you're gonna set up, then you'll be next. Do you want? You can download your presentation on mine.
Yeah, can I just have him set it up? I can't stay much longer.
Okay, so there you go.
Can I take this to my seat for a
Yep.
sec?
No, we're done.
We're
good to go. Ready to rock. Can't wait to
No
see what you have to
pressure.
say. Do you want to clicker? You want me to click for you?
That'd be... If you can hook up, that'd be awesome.
No, I don't know where it went.
Oh, okay. Then no, I'll just...
Did you get your clicker back? It's gone. Here it is.
Awesome.
Okay, just go ahead and do that.
That's weird.
Okay, there you go.
The ball's spinning. So hi, I'm Amanda Parks, and I'm also a member of this space, and I'm also a member of Gen Space. And I'm originally a product designer and an exhibition designer, and then I ended up at the Media Lab in the same group as James, doing tangible media, and I did a PhD. Oh man, I'm gonna cry. I did a PhD in physical computing, mostly working on kinetic interaction, but in the middle of it I got very involved in different kinds of biological ideas. And what you're looking at is a vat of algae. I'm going to keep talking until the ball stops spinning. What you're looking at is a vat of algae. And there we go. And I own, right now, I'm the founder and the CTO of Bodega Algae, which is an algae to biofuels company. And I think the reason that I'm sort of in this space is what?
There you go.
Okay.
Oh, you want to set different?
No, someone just. That's not.
Oh, that's yours.
It is mine. Okay. Oh, it did like. Okay, it's a second screen thing.
Yeah.
Yeah, just wait. Okay, there we go. This is like really complicated. See what happens. I lost my train of thought. Anyway, so I am working on like a real commercial company, but I'm also doing artistic exploration with algae. And basically how this happened was, as part of my sort of graduate student work, I was doing research in sustainable architecture, and I was looking at kind of the potentials of algae as a biofuel, and a friend of mine who's an optical engineer, late at night in a bar, we're talking, and he's like, oh, there's actually this historical research at NASA looking at how you can use the flashing light effect and different kinds of optical technologies, which he was working on in the flat panel display industry. We kind of teamed up and started thinking about how we could actually start to spread light into algae volumes. The reason that that's important is that basically when algae starts to grow, as it becomes denser and denser, it starts to self-shade. And if the algae can't actually get light in the bottom, it actually chemically signals the rest of the cells in the batch to stop dividing, so it sort of kills itself. So this idea of having light distribution in large volumes of water was very important. So we went around and we were doing research, and we went to several different labs, and I fell in love with algae as a design material, like kind of thinking, this is great, this has the potential to be fuel, but I want to see it in domestic environments and urban environments, and I wanted to see it used as a material in the same way that kind of aquariums are put in for different kinds of calming psychological effects, and I fell in love with the color of it. And I started to imagine, scenarios like this, where you could have these sorts of growing columns where you could just charge yourself out at the bottom. There could be the whole processing, sort of, embedded. And this was like in 2007, before this, like, there's been a lot more talk of algae recently in the last couple of years. Also thinking about kind of having it as a wall in your home, and it would just feed right into the sort of central stove heating system. So why is algae good as a biofuel? it's a lot about sort of how much you can get per acre of land. That's an auto-advance thing. And compared to other kinds of biomass substrates that can become biodiesel, macroalgae is sort of orders of magnitude higher in terms of its sort of yield. If anyone has questions, I'll go into this. I'm trying to just cut everything super short. The way that algae is normally grown right now is in pond systems, closed systems, and open systems. Ponds and raceways are open. Closed photobioreactors are, the issue with the closed reactors is that you end up with very, a huge number of small containers, but it's all about surface area to volume ratio. How can the algae get light? When you're in a pond system, you're basically trying to move the algae around as much as you can to get it to the surface to get light. That's really not effective, and it also, you can have shear strain on the algae and kill it. And then inside of the closed photobioreactors, you're basically looking at a harvesting problem, where how do you control and harvest these sort of literally millions of plastic bags and try to empty them to make oil. So our systems are these modular, scalable photobioreactors, which are kind of, we're working on this series of different sizes. And this is a meter by meter by meter, which is the smallest size, and sort of up sort of the largest scale that we're going to a laboratory. And we're working on dumpster-sized reactors, which we're doing some kind of experiments with how do we sort of place these into different carbon CO2 remediation sites, and then going up to sort of larger scale photobioreactors, which will be placed on coal-fired power plants or biomass-fired power plants that kind of co-burn. So we're kind of going up in these three different scales. But basically the basis of our technology is not really about trying to become a sort of industrial power plant manufacturer or anything scary like that, because I can't even get my head around that. It's about the optical technology, and it's spreading light into the volumes of water through these massive, cheap optics, which are these lenses that you see inside here. And so yeah, basically what happens is this is the sort of money shot for the investors, where there's light passing into these lenses, which it's all about total internal reflection inside the lens, the angle at which the light goes in, the angle at which the lens is cut as a wedge, and then all the surface finishes and very kind of detailed stuff around that. But the tank on the right is our technology. We can see it's totally equally lit, and then the tank on the left is without it. Same amount of light. And then we also have a system that flashes either solar or artificial light at very high frequencies, which has been shown to actually grow the algae more effectively in terms of, it doesn't oversaturate in terms of photosynthesis. So you can actually use less light to grow it at the same effective rate. People ask me about the whole kind of start to finish cycle with algae. We're working just only in the middle section, but what you do is you can feed it waste and CO2, which is great. And actually a lot of the models, the economic models around this right now are just about treating wastewater and treating CO2. We're not even at effective densities to compete as a fuel yet. But those are the sort of inputs. Then you get free solar energy, or you can also use sort of light that is energy that's coming off of power plants, excess energy, and you can use artificial light at different times, if that's kind of makes sense in terms of the economics. Extraction process, either through a chemical or mechanical or potentially a viral process, where the virus actually is co-grown with the algae, and when algae dies, it self-lies the cells and then extracts the oil naturally. And then the kinds of, what are the markets that we're sort of going after? Or what are what's funny is that the algae that's in baby food, or the algae that's fed to fish, and the algae that's used as fuel, it's all the same thing. Kind of economics of scale about the price point of it. There's like, outside of, potentially some really damaging wastewater chemicals being its feed, there's nothing else different about it. One project we've been investigating is working with a landfill landfill gas to power plant. This is a situation where you have a landfill that collects the methane and then burns the methane in these special engines, and then they use the excess heat from these engines to heat greenhouses where they grow hydroponic tomatoes and sell directly to Whole Foods, literally on top of the landfill. Amazing place. And they have an extra greenhouse, which we're talking to them about using, and we would try to sink their CO2 and carbon monoxide. We're hung up on the carbon monoxide calculations because algae just doesn't eat it quickly enough for them to care. So it's all sort of economics, government policy. When someone asks me, how much how much does a gallon of algae cost, or how much can you grow, I say, what state are you in? What are the incentives? It's not a sort of direct correlation in any way. It's completely political. So getting to the art part, this is what we're working on right now, the second generation of the tanks, which are continuous flow photobioreactors. What you saw before were batch photobioreactors, which means that you put in an inoculation culture, you let it grow for the cycle is basically 2 weeks. You see what kinds of densities you get with a set amount of food and a set amount of light. This is a continuously harvesting system where you have food, basically this media makeup, continuously going in, and you have you're continuously harvesting, and you're trying to keep it at its peak growth rate, its peak density, and then there's just a lot of sort of sensing and filtering around that. But what I'm trying to do with this as prototype for this is I'm working on an algae pixel display, going back to the kind of art part of this. I'm also a scientist in residence at NYU at DP right now, and I teach also in architecture program at Columbia, so just kind of research, joint research with that. And so I'm basically using these little boxes as kind of pixels, small, very, very small continuous flow cubes as a pixel display. And then what you can actually do with the algae is map. I have a Pantone mapping of like day 1 to day 12. And so you get this kind of monochromatic display possibility. So when you think about going up, you know, of course it's going to be super low res right now. I'm working on about 12 pixels. But you can think about it as a real kind of potentially like biological organic pixel. And in a sense, it's interesting to me from the concept of slow media and displays that are the exact opposite of what we think in Times Square, where everything changes continuously. But this can actually be about, you know, the environment and about what's around it. And in a sense, you know, if you start talking about, okay, we have to wait for it to grow to get to that darkest color. You know, you can remove the algae quickly and get to a lighter color, but if you have to, so you have to build these algorithms around waiting for certain things to happen. Which I mean, I think that's just a different kind of interesting way to think about conveying information. Computation. That's it. Thanks.
What kind of algae are there?
Well, there's a bunch of different kinds. Right now we're using Chlorella sorkiniana, which is like a kind of generic, not generic, but it's a strain that is kind of easy to grow. It grows relatively well in conditions that are not entirely sterile, which is something that we want if we're working on something industrial. It grows relatively quickly, but with a high lipid content, so it's like a balance of all those things. But
are you going to stick around over the weekend?
Well, I have algae in there in GenSpace. If you want to get it out, you guys are welcome to use it. But I can come in and out. I can't be here all weekend. But.
Well, if you have any interesting projects to work on, take advantage of them.
Yeah, I mean, if anyone wants to play around with like the pixel ideas, you know,
I certainly do, and I brought some algae too.
Yeah.
So like maybe.
Right. Yeah. You definitely should have me talk. So. Yeah.
Cool.
I mean, if anyone wants to play around with
So you want to present the that was mine actually. That's.
I just have the websites up here now. So
Okay,
should I just
great.
use this?
I do. Good.
Food is here.
You're in a browser?
Yeah.
So you can pick the browser window and drag it.
Maybe you have like two screens and you do the arrangement thing.
Yeah, I'm trying to do it, but I'm not mirroring.
Got the windows, mirror display.
Yeah, that's what I did. I'm mirror it now. I guess it's just the wrong resolution.
That's good though.
That's not the resolution of the screen
Not as good as
Well, I need the screen up there, so what do you want me to do? They're on the Okay, well, so this is far from perfect, but I don't know. Waste more time. I'm Beatrice. So I thought I would talk of about one of the latest projects I did, but I realized I should maybe mention at the end some of the wacko work I've done in the past, since it seems to fit this context a little better. But last year what I developed was, speaking of iPhone apps, it came out earlier, there was a time when we all built or had to build our own hardware for more localized technologies, things I've been involved in the past. And last year for the first time I was like, okay, I go with a mass device that's being deployed, people have it, I'm not going to build my own hardware and see how that goes in terms of political intervention. And on top of it, as the platform, which is an iPhone, which of course is problematic since it's closed. So the project we developed, I called it the Endangered Species Finder, and the idea was that it would be an iPhone app that provides users with the ability to locate habitats of species that are normally under threat, and that was specific to the United Kingdom because that's where it was from. So what you see here is just the database of species that we had from the UK, and we mostly went with, we only went with terrestrial and sweetwater species. So we didn't include the entire ocean around what sort of counts as territory of the UK, since the database basically would have exploded. So from this, from just this menu, you would go to like the specific species. In this case, it's the common toad. Believe it or not, the common toad is endangered. And you would find more information about that particular animal in this case. So information about the habitat and ecology, the reasons for endangerment, breeding habits, and markers which would help you to recognize if you're indeed in such an area where this animal might occur. And we were this screen doesn't help. And then you could locate yourself on the map in relationship to those known habitats, which in the case of the common toad, it includes the entire UK, because the way endangerment listings work is that some of them are really specific to one territory, so to one geographical area, and others are simply rare at this point or decreasing in trend to an extent that they're listed as being endangered, but you technically could find them anywhere, which is why we had to give a description of the visual markers. So if you were inside a building on the 5th floor, you would kind of understand you're probably not going to encounter a common toad right there. But the idea for me was really that I think a lot of our ethical behavior towards environmental issues, some of them come from policy, some of them just come from what we're trained or culturally brought up to believe. But I think a lot really only occurs once you encounter a certain species. So it's not that I will build a personal relationship to the mosquitoes on my front porch, most likely. But if I've never seen a common toad, it's kind of hard to develop any type of opinion about its endangerment or potential extinction. So what I was hoping, like, what a lot of technology does is providing for species for us to think differently or look differently. So in this case, it was really meant as a device that would encourage you to look around. Am I actually seeing this, what this device is telling me I might be seeing if I would want to encounter this particular species or not. And then we also implemented, so if you actually had a sighting, you actually saw one of those, that you could submit it, like send us an email, and we could trace what people might have seen or not have seen, because the overall goal eventually was a collaborative website where we would collect these sightings. But this time, you probably, load of you have been in the same situation. Once you design systems that entirely depend on user participation, but if you don't get users, you design a big fat system that kind of sits there unused and 2 years later it's out of date. So in this case, I wanted to make sure that actually real participation before I build a huge online process. The next version of this one will be in Android, and it will be for the United States, and for the entire US, not specific to the UK. And we'll be working with people at Stanford from the Center for the American West to develop interest in this particular project, but just an environmental visualization on small devices in general. So I'm excited about that, because this one was very much, it actually took a lot of research. So on the database, on the website, if you click the different parts, you will get all the source information, the distribution of the species, and whatever we found out we can learn about them. But with an institution, in this case, like a huge university and researchers behind me, I think I can be a little bit more in a facilitating role for handling this large amount of data, because I know some programming, but not to that extent. So, and that project was developed in conjunction with an exhibit, so my background is in visual art, in London, where I worked with collections from the Natural History Museum in London and the Horniman Museum, which is also a science museum in London. And these are all specimens of endangered species endemic to the UK. And essentially what we gave these species is a birth date, which is the date that were recorded officially by the various naturalists at the time into Western science. So we will have a lot of lineage 1758 as a date, and a projected extinction date, which in most cases is simply question mark, because it's not something you can easily, or in most cases not at all, predict. But for me here, it was really kind of showing that the only mode encounter that will be left once a specimen, a species really is extinct, is in this kind of museological research relationship. So you either have the gaze of the visitor in the museum, or you have the gaze of the scientist, but you don't have any living encounter. So to develop a relationship at that point is very, very restrained. So I think for this context, what I don't have time to talk about my 5 minutes that I probably already have used up, but a lot of my previous work was more dealing with wetware on a smaller scale. So 2 years ago, I developed a project I called Invisible Earthlings, where I worked with microbes. And here I was really interested in kind of my invisible neighbors, so all the creatures that live around me that I have no idea that are there. So I literally took samples from my backyard and did some microscopy to analyze the molds and kind of look at the cultural relationships between humans and those microbes. So I had a lot of penicillium in my backyard, which one species is used obviously originally to derive penicillin, but a different one is also used to make camembert. So I kind of was tracing these kinds of relationships because I think, especially when it comes to microbes, we have primarily negative associations because that's when we have to think about them. When our walls kind of develop molds or when we get sick, right? But a lot of the other kind of ecological and cultural relationships we might have with these microbes, we tend to not be thinking about. This one I actually did some, yeah, I'm done. Okay, so there's some more about synthetic biology on here. There's some stuff about pigeons and air pollution levels. In the bottom, you have transgenic bacteria mixed with human genome. You have an open public lab where we tested food crops in Europe 8 years ago. So the whole idea of an open lab, I'm really excited about because you guys are kind of doing what we are thinking about and we could only do on a time-lapse basis. So thank you.
We'll have to come back to your website during the weekend because it seems like there's a lot of great launching off points for. You should maybe elaborate on some of your projects that exist already. Yeah, that's up. You're up. Yeah, Rich.
I want to try out the clicker thing. Can I try the clicker?
Yeah, you sure can.
Yeah, but then it's this one. I don't know. We have to make.
Can you pass the USB to me?
Yeah. Okay, this is.
It just seems to take a second to initialize. Just go to cool, I think it works.
All right.
And it's still not up.
Right. Well, that's that's another thing. Next. Okay. Need a clicker. Yeah. Oh, it works. Right. Okay.
So my name is Rich, and I am here representing a group called the Center for Post-Natural History. This organization's been kind of in the process of becoming for about 6 years now. It has been in existence in Maine for about 3. It basically came about out of kind of an interest in synthetic biology and at the same time sort of evolutionary biology, and an awareness that there was this kind of missing piece that wasn't being represented in natural history museums. And the more I dug into it, the reasons kind of why there was this sort of blind spot, why organisms that are modified by people aren't represented in the Natural History Museum became this kind of nagging issue, preserving of perhaps its own word. What we're fundamentally interested in is this overlap between nature and culture, where culture, kind of the cultural footprint exists in like the heritable biome. So, what does that mean? For example, in the all-inclusive sense of the word post-natural, we're talking about everything from the dawn of agriculture, the domestication of animals,
all the way through to synthetic biology and whatever it comes next. Interesting footnote, these are the work of selective breeding. These are not transgenic. So this is kind of new school meets old school in a way. So towards this end, one slide, it's a nagging slide. All right, well,
I got it.
well, if you can just remember that slide that we passed over a few times. I don't know why it's sticky. But we do have a storefront space in Pittsburgh, Pennsylvania. It's officially opening in a week, 4913 Penn Ave. Website, postnatural.org is where you can follow us. We maintain a collection of living specimens whenever legally possible, which is most of the time not, right? Especially when it comes to transgenic organisms. There's very few that a person like myself is able to legally have outside of a laboratory space. The things that I am legally allowed to have are kind of the interesting exceptions to the rules. So you've got the blowfish, the only transgenic vertebrate that people like us are allowed to have as a pet. But then you've got Monsanto Roundup Ready corn, which I'm not because I didn't sign a contract with them, right? I'm not really, it's just like, maybe there's a mirror. It's done it twice. So for instance, this Roundup Ready corn, I was able to recover from commercially sold birdseed, and which I just planted the corn that was in it, grew it up, cross-selected it with Roundup, and this stuff will stay growing. So this is a way that I was able to more or less legally acquire Roundup Ready corn for exhibition purposes. But the point is, most of the stuff that I get...
Did you test it, Rich? Did you like make sure that it was transgenic by sequencing it?
Well, I had a lot of control groups, and they died within...
Oh, so you put Roundup on it?
Yeah, like I cross-selected with Roundup. It works great. Try it. So most of the specimens in the collection are necessarily dead. And the interesting kind of thing I discovered was that I'm allowed to legally have anything as long as it's dead. And so far I've never been turned away by a researcher when I approached them and said, hey, I would like to collect your specimens for our collection. Can I come to your lab and kill some and take them with me? That's totally okay. Sometimes there's things that we can't get access to and we revert to the old standby diorama. In this case, this is a Defense Department goat herd that they're literally raising in former nuclear weapons bunkers in the old Plattsburgh Air Force Base. So what I'm here to talk about today is a notion of what I call permitted habitats. So when we're talking about, and this is specific to genetically modified or transgenic organisms, their habitat isn't defined by, say, ecology, the way an organism in natural history is going to be. Their habitat is defined by policy. It's defined at the national level and the state level. And so when it comes to tracking these organisms,
I'll pick you up.
for example, the Transgenic American Chestnut Tree Project. This is happening at the Environmental Science and Forestry Program at Syracuse, New York. So these are chestnut trees that are being engineered to be resistant to the blight that's killed off, more or less, the American chestnut tree, or just kind of reduced it to a permanently stunted shrub. They're grown from culture, and their habitat is defined by
a large smiley face.
The large smiley face. This is the signifier that says, "I am transgenic. Don't let me leave the building." And so the smiley faces follow them throughout their entire lives as these kind of benevolent angels of death to let them know that they will never leave. With the exception of this case. Right? So these are the only grove of transgenic trees in a publicly accessible place in North America. This is just outside the library. And they had to go through quite a few hoops in order to be able to do this. The regulatory process is quite thorough. In fact, it was mandated that they had to be both fenced in and had to have night vision surveillance cameras. So, when it comes to this project of tracking and exhibiting the genetically modified world, which is the project that we've taken on, it's a real question: how do we know what's out there? There is no central repository for this knowledge, and if you just go by, say, press releases, well, then you're going to the source that we generally start from is a database called the Animal and Plant Health Inspection Service, APHIS. It's part of the USDA. It's something that you can check out. And within it, every time a company or university wants to take a genetically modified organism outside of the lab, they have to file for a field release permit. So that's what these all are. This happens to be DuPont. These are all corn and soybean, right? It's corn, soybean, alfalfa. Those are kind of the primary crops. These are the genes that are added to this particular one. And interestingly, they even tell us where they come from, which is, as a post-natural historian, that is interesting. It tells us a little bit about what they might do. And then lastly, where the permit is being issued for. And in this case, Hawaii and Puerto Rico. We'll get to that in a minute. There's also quite often fields that are labeled CBI, which stands for confidential business information. This means this information has been redacted. And so throughout the entire database, CBI shows up as a species, as a location, as a gene. And so we end up having to necessarily build exhibits of CBI as all of those organisms. And you can never exhibit exactly what they are because you don't really know. This is tricky, but I want to hit the mouse button. Somebody put I don't know, just hit let's try again. Okay. Wait, one more time. Wiggle the pad around and then click. Right? That's one click. Sweet. Okay. So this is a visualization of that same database, starting in 1987 all the way up until last year. So there's a date up there, and it's going month by month. This is every field release permit, an approximation, an icon for the species that was given to and the location. So this gives you just kind of a broad bird's eye view of kind of what's gone down in the last 20 years or so. You start to see the Iowa Corn Belt exploding. There's this weird thing happening over here. So this is the edge of the continental U.S. This is Hawaii. Yeah, it's supposed to be over there. And that's Puerto Rico down there. They're going crazy with the corn, which is weird. You don't think of Hawaii as being part of the Corn Belt. But it turns out that during the last 5 or 6 years, corn has become the primary cash crop in Hawaii. Now, it's not corn that we eat, and it's not corn that is fed to cattle. It's the experimental parent seed. So these are closed fields that all the companies, Syngenta, Monsanto, Bayer, Dow, DuPont, they're all buying up huge fields in Hawaii for the purposes of raising their parent seed, this is the very upstream in the process of experimental corn. So here's an example of a permit that was issued to Monsanto for corn. This is all the genes that went into this one species of corn. These are all CBI. It tells us where they came from, which is interesting. Right? From that information, you can kind of, if we are a little bit familiar with the organisms, we can take some guesses as to what they're interested in. But they're kind of interested in everything, as you can see. So, and again, Hawaii and Puerto Rico is the destination. So what's that all about? Well, Hawaii. If we kind of hover over Hawaii, if we hover between like Google Earth and Googling addresses of these companies, we can start to look kind of peer over the fence and get an idea of what's going on where. So this is on Hawaii. This is an old sugarcane field, I believe. The sugarcane industry left Hawaii in like 99, 98, but corn stepped in. And so these light patches are the test fields. It's kind of interesting, they're like very isolated from one another necessarily, so they have to be. They're not allowed to cross-pollinate. This particular field I found based on a hunting permit. It was given for everything north of the DuPont hybrid fields. Otherwise, it didn't identify itself. So that was a handy piece of information. This is a particularly interesting valley. This is a valley called Cunha on the island of Oahu. Monsanto, Syngenta, and Bayer have all bought out this field. That might not be interesting, not from the fact that I was trying to figure out how I could go there and document it. I found out that the airspace is closed up to 19,000 feet. What would that mean? Well, it's a really interesting neighborhood, as it turns out. You've got Wheeler Air Force Base, Schofield Army Barracks, and a mountain range that's closed military space that spills over into a Navy munitions bunker field, and an underground NSA listening site, which is literally underneath the cornfields. So if you ever wondered what an NSA listening site might look like, well, imagine if you had a building about the size of a 7-Eleven, and then you parked 3,000 cars outside.
With a helipad.
With a helipad. So that is a comically large number of people showing up to work here. I think so. So necessarily, these places are closed estates. So from the perspective of a post-natural historian, how do you document that? Photography is one. From a specimen collection problem, this becomes even trickier. Knowing what it is that you're looking at could benefit from some of the stuff that Mac was talking about. If you kind of knew what you were looking for. And why would you care? Well, so Roundup has been kind of in use for a long time now, long enough that its use is starting to wane as weeds become Roundup resistant when you go to glufosinate, which is actually a different company that owns that patent. So Monsanto is not going to go with that. You eventually start to run into old-fashioned defoliants like Agent Orange, and Dow has patents on those as well. So I would really be interested in looking for evidence of crops that are resistant to Agent Orange, which I think would be pretty popular. So there's a million other things I could talk about, but I just wanted to focus on that one. If anybody's interested in like doing a little cold searching in these databases, it's really fun to start off like with the place that you live and things you know. The things that come out are, it's kind of a fractal. You can follow them down these little different rabbit holes and you never know what you're going to find. I've got a bunch of like brochures and stuff like that too. So, thanks.
Pretty fun. Should we do a little break?
I think there's only 2 more talks. There's Bree and there's you. Oh wait, and Mark. And Mark. So I can, yes, I will be a taskmaster. You will all have 5 minutes, and I will cut you off. So far, 5 minutes has really meant more like 10 minutes, so that's why it's now later than we were hoping. I couldn't, in good conscience, cut you off because you were on fire. It was really fun to listen to. But you can have this back. And we're going to get through it. And I know it's getting late, but I was kind of hoping that post-talks, we could have an informal brainstorming session, and the way it might work is, let me just try it right now. Did anyone come here with an idea for a project? I did. Anyone else? 1. No one else has any ideas for something like that? 2 people? Do you have anything? Rich, do you have anything? Where did Rich go?
I'm over here.
You don't have any ideas for something to work on? You ended your talk with an idea, which was we can look at some databases together.
Okay, well that I can.
Right, yeah, so like that's the sort of level I'm talking about for an idea. So who has that kind of idea?
I have a device I'd like to build.
4. So 4 people. And we were kind of hoping that Future Lab Camp would have like 5 teams. So we're only missing 1 idea, or 1 pitch. So after the talks, I'm hoping that the 4 people who raised their hands could pitch an idea, and whoever finds that interesting goes stand by that person, and we will see how many people are left over, and those people can participate in a brainstorming session with me to see if they want to work on a project, if we can come up with a project. And the goal is to leave the night with everybody who's going to participate in the workshop with a project that they can discard tomorrow, but if they don't want to, they don't have to. That might be a 10-minute process. So is everyone up for that? Okay, good. Then it will be so.
So I'm Mark Siegel. I'm an evolutionary biologist from NYU, and I'm going to tell you about some cool biology. We're excited. It's a project that we're on with Sasha. So the topic is survival in an uncertain environment. And just to relate it to something you're probably familiar with, that says diversification to reduce financial risk. So as you probably know, if you don't want all of your money to disappear, the best strategy is to have a diversified investment portfolio. And the Securities and Exchange Commission, when they're not busy drawing pyramid schemes, will educate consumers about how to do And so it shouldn't surprise you then that because evolution cares about long-term returns, that organisms also have diversified portfolios and hedge their bets in a real sense that resembles investing. So to give you an example, this is, sorry, that gets cut off. That's percent of seeds germinating of this giant ragweed. Okay, and so ragweed is obviously a weed. And what you'll notice about when, in terms of the day of the year that the seeds germinate, it doesn't all happen at once. And in fact, there's two peaks of germination, one at about day 100 and one at about day 150. And so those are two sort of investment strategies. It could be that on day 100 conditions are good and that's the best time for it to germinate, or it could be on day 150. They don't really know ahead of time and sort of spread out the risk. Contrast that with commercial canola, which is obviously planted in huge monoculture in lots of places, and it will germinate, almost all seeds will germinate within 2 weeks of planting. And that's because, to every extent possible, the farmer or the agribusiness has controlled the environment. So there's no longer any risk. There's plenty of fertilizer, there's plenty of pesticides, there's plenty of water, and so there's no need for canola to hedge its bets. And in fact, the best investment strategy when you know you're going to win is to just put all your eggs in the same basket.
But Mark, when you plant the ragweed, you plant it on day 0, and then it might be 100 days later that it grows, or it might be 150. It might be, you don't know for every
so
seed.
you're not planting ragweed.
It's just growing. Oh, I see. Okay.
So one of the organisms we study in my lab is budding yeast. I brought some with me from my chair. So these guys are cool. They're microbes. They're about 5 millions of a liter across. One of their neat features, which will come up later, you can see them budding. They're called budding yeast because when they divide, they come into little buds. Every time they bud, they leave a scar. So you can look at them and tell how old they are. You just count up how many scars and you know how many self-limits they come through. And so what we wanted to see was whether budding yeast had a diversified investment strategy. And so the reason we wanted to use budding yeast is because we can do genetics, we can do molecular biology, and hope to find out how the organism actually functions. So what we use is this device. It's a Nikon microscope. If someone could build me one of these for $100, that would be great. This would cost $90,000. It's basically an environmental chamber sitting on top of a microscope, and it's fully automated. So the stage, the filters, the optics, all moved by computer. And what that allows us to do is watch a lot of cells grow. And so this is what it looks like. On the left, you'll see a field of yeast cells growing. On the right, you'll see our custom software that tracks each of these little rolling colonies. Okay, so you can see these colonies forming, and our software can track them and measure the area of the colony as it increases. And if I do a blow up and focus on 4 colonies as they grow, you'll notice something very peculiar about these colonies. And that is that despite being genetically identical, despite being in perfect conditions, they're growing at vastly different rates. So this colony is exploding while this colony is just sort of hanging out. And when I saw this, I said, this is bedhead. And what we needed to look for then was a correlation between how fast these colonies were growing and how well they can deal with some unpredictable stress. And so we can do that experiment. And we can do even better than that. Because by using genetics, we were actually able to identify a gene that correlates with this sort of behavior. And so this is a time-lapse image of 2 colonies growing. And you'll see that some of the cells are green. That's because they've activated this particular gene. And we've engineered them so that they make green fluorescent protein instead of the normal protein. And so what you can see if you look at this colony is it's not very green and it's growing very fast. Whereas if you look at this colony, it's green from the beginning, and it's growing very slow. And we can look at 100,000 colonies like this, and we see this correlation. The greener it is, the more likely it is to be growing slowly. So that gives us a handle on the genetic basis of this slow growth and what it might do for the organism. And so the kicker experiment is is then to grow these cells for a while, and then stress them and see who survives. Because if this really is a hedging investment strategy, then there not only should be this variation in how they grow, but that should do something, and maybe these slow guys are the ones that can survive better after stress. And so what I'm going to show you is a movie. Over here we have just the images of the cells growing. Over here is that green isolated in some channel. So you see this, see that sort of white flashes over here. And so I'm going to play it, and then hopefully I'll be able to go back and play it again and point something out to you. So what you're going to see is the cells are growing, then it's going to flash heat shock. So that we're going to give them a very intense heat treatment that they usually can't survive. And then we're going to let them grow again. So I'm going to play it again, and so the action is all here, right? So this is the guy that survives the heat shock. Those guys all dying. And if you watch it again, and you pay attention to which one's green. I'm sorry. Yeah, green guys, slow growing, and then they're the ones that survive the heat shock. Okay? So that strategy of growing slowly, even though times are good, is actually a good one when times get bad. To finish up, we noticed something really interesting about these cells. As I said, you can tell how old a yeast cell is by counting how many scars it has. You can do that with some people too, I guess. And so this graph just shows that the slower growing, if you compare the slower growing, the green cell, to the faster growing cell, the slower growing cells are on average older. They have more blood scars than the fast growing cells. And so this, I think, is something quite profound because I think you normally would want to say, well, they're older, so they're slower. Okay? But what I just showed you is that those slow ones were actually the ones that were not just going quietly into the night, but they were actually taking a risk so that when conditions get bad, they're the ones that are actually going to be surviving. Okay, so they're taking a calculated gamble in old age to do something for the good of the population. And so what I'm going to leave you with is a quote from the philosopher Robert Nozick, which I think sort of holds this result. And that is, he said that, that some might seriously weigh spending their last years in a brave and noble endeavor to benefit others, not going gentle into everything or raging against the dying of life. And near the end, shining their light on spite of the world.
That's great. So are you interested in cheap microscopes? Is that the device? So the neat thing is that there's a design out there, an open source hardware design for a 2-axis stage actuated by Arduinos and servos, cheap servos. And I'm not sure that the steps that it can move in 2 axes is good enough to drive around and look at yeast. And the webcams that I brought, also when you invert magnification. Yeah, 100x is not quite good enough, right? You need more like 400 or something. We use like 200, right? So maybe we could get there. So I feel like it's almost good enough to do like a $1,000 machine would be trivial. Not trivial, but like if you had $1,000, you could probably do it. Really? Why would you be 90 though? Yeah, exactly. And what kind of software are you using to do the analysis?
So it's a mix of MATLAB and R.
So it's totally
the
homebrew.
software language from Nikon.
Okay. Cool. I don't remember the name of it, but there's a open source microscope control library and software or an image analysis package.
Yeah, one of the reasons we got the Nikon is that some of the other makers like to will not tell you what their input output protocol are. But Nikon does, so that
you could move in that direction.
Open source.
Cool. Bree, yeah. So I think Bree is going next. Bree? Okay, cool. You're there already. Awesome. So yeah. Does that work?
I think now you're on.
Okay,
There was a
physical
clicker.
matchups.
This is the clicker.
Yeah.
Oh, great. We plugged it in.
Super.
Yeah, it does work.
Super.
Awesome.
There's a button I push. Great. Okay, so yeah, let's talk about taking things and putting them together. It might seem kind of like Play-Doh, something like this, but I hope it's not really more interesting. It really starts here with the Gangsta by Wyzord, or Zord, some people might say. It's a very boring model. It's a guy wearing a hoodie, with his hands in the hoodie. And there's not actually a lot not a lot of detail. Very smoothed over, boring model. In fact, it's so boring. Oh, I'm giving it all away. Then you have a model that might also look boring. It's a snake. Well, a skeleton of a snake. And then you put them together, and you get snake stuff. It's a gangster plus a snake. It's snake stuff. Now all of a sudden it gets more interesting. Like, okay, it's a snake gangster. I want one of these. And then somebody takes that gangster and they're like, okay, if we're playing with the gangster, I'm going to make it a low-res gangster. Low-res stuff. I'm going to put a devil head on him, and it's going to be devil stuff. I'm going to make it a talisman by putting a hook on his head and putting Medusa on its chest and some creepy face. Well, Stella stuff. Let's remix it with Buddha and call it Bood stuff. Let's remix Bood stuff and Stella stuff to get Bood stuff the legend continues. And then, of course, there's Stanford Bunny. You see what's doing? For those of you who are not obsessed with 3D models, the Stanford Bunny is something like 20 years ago. Literally, one of the guys at Stanford got a bunny at a thrift store that was made of ceramic.
Oh, what's that? All right.
Shake it. And they scanned it. And the idea was to make a really massive, like, lots of points. Like, what?
So the Stanford Bunny is really from a thrift store somewhere?
Yeah.
It was like some ceramic thing.
But they totally tried to, and they actually have a big long thing on their site that says, like, we're protecting the points of this bunny, this Stanford property, but nobody's taking it.
Really?
Yes.
Oh my god.
So of course there's the rabbit stuff, which is the rabbit head on the gangsta, or you can take the gangsta head and body and put it on the bunny. And then somebody took the bunny and, of course, decimated it, which makes it all triangles, and made the Tron bunny. And then somebody took Optimus Prime's head and put it on the bunny, so it's Optimus Bunnius. And then some Primesta, Optimus Prime on the gangsta. And then, of course, you've got the Lepus Draconis, the rabbit, inside joke. Then there's, giving it away, Han Solo. Carbonite, of course. You've got carbonite stuff. It just keeps going until you get to Sta, which is you take Sta and you cut him in half and you put it on itself. So you just so then there's Walt Disney's head. This is a model that's really old. This guy who worked at Disney ended up hating Disney and doing this whole video sequence where he cut off Disney's head and put it on a plate. And then he took that really realistic model and he just gave it away Creative Commons, which is fantastic. So everybody prints out Disney's head. And then, of course, there's this guy, J-Mill, who does a bunch of academic research. And this is an academic research brain that you can just download. So he's like, yo, this brain, print it out. And he's like, but put it in Disney's head. And then, of course, somebody says, okay, we've got the gangsta, let's make it P. So let's have Pista. And then there's a urinal somebody puts up just because, you know.
It's some kind of art tradition.
Yeah. And then, of course, somebody,
it's the standard
got a flushable toilet now. So it really does, it's a model. It's a model flushing. And then his and her toilets. And then you end up with grainy Walt Disney stuff, which is Yeah. And then we go, oh, we've got brainy Walt Disney stuff being in Paramount. And then this isn't even my quote. This is brainy Walt Disney stuff's quote. I hope we never lose sight of one fact. This is also by gangsta. So, this is just a short, this is what I'm interested in today. I made this thing for you because I think it's interesting. And it's happening right now. This whole like people, and this is just one thing about mashups with stuff. I mean, there's a whole other thing where I got a really high-res scan of my head and I uploaded that. I'm not even touching that because it's pornographic. People put my head on everything. Please try it. And so this is really easy to take something, put them together, win. And you can use, like, really, there's lots of free options for you. So Blender, SketchUp, all these kind of things. There's also this really nice one, OpenSCAD if you're a programmer. This is like basically you say like, hey, I'm going to put these two things together to make this. I'm going to add this, and I'm going to add this. Oh, and then I'm going to take this and put it with this, but subtract all the parts that are left on the outside of that. And then I'm going to put these two together and do a Boolean operation and end up with that. Boolean means that you subtract one thing from the other spatially. And then there's this thing called Mesh Mixer, which is just a disaster waiting to happen. And they just relaunched another one. Now you can use it on the OS X, which is nice. And you literally take like a person and a horse, and you can cut them in parts and stick them next to each other and stuff like that. Take the ears off an elephant and the face, and you're good to go. And one of the nice things is a culture of derivatives and a culture of sharing makes this happen. Really a culture of sharing, not derivatives. I'll change this one. But you know, public domain, GPL, Creative Commons, basically publicly saying, yo, mess with my stuff, makes a ton of really cool stuff happen. You might have the next gangsta that would inspire other people to do really stupid things. So, share it. That's it.
Cool. Chris, you're up.
All right. So since the topic is about like inventing prototyping with biological features, I thought I want to talk about like prototyping futures and also trying to do. I just did this presentation. It's very freestyle. So. So let me see. I have to be here, sorry, because this is all right. It's very messy. So let me start off with okay. Let me start off with this is a video compilation that I did actually like a little while ago. It's compiling like future vision of like corporate visions of the future from like Silicon Valley companies, and it starts off like 1984. This is AT&T, Ameritech, formerly Ameritech, talking about okay, we have like sound system here.
Current regulations bar the company's involvement. One future offering is electronic yellow pages. If you think of our business and the fact that we have telephone directories and advertising appearing in these directories, we can someday put that advertising into a database. And then people sitting at home actually have the ability to dial into that database and, similar to paging to a directory, retrieve information from it. The next nature of our business is trying to bring together consumers and advertisers. Let's just take some potential applications. Let's assume that you're sitting at home, that you peruse a menu, and it's to your liking.
Here's my sauce. We have a black ravioli made from a cuttlefish and a seafood and a shrimp sauce.
And key information back and forth.
So it's a compilation of well, this is Apple Knowledge Navigator. Oh, this should release the audio.
All universities with geography notes.
And what happened to the sound now? But what we what I'm really interested is in the...
Copy the last 30 years at this location at 1 month intervals.
Excuse me.
The
Jill
aesthetics
Gilbert
and
is
the
calling back.
scenarios and how the futures are communicated.
Hi, Mike. What's up? Jill, thanks for getting back to me. Well, I guess that new grant of yours hasn't dampened your data learning abilities. Rumor has it that you just put out the definitive article on deforestation.
Aha.
So this is basically the
Is this one
iPad,
of your
like from
typical
like
last-minute
vision of
panics
like
with
1984.
No, that's not until...
But the environment is kind of interesting how they imagine the future. We can see later. This is like kind of the future home with like, you know, the library and like this old style like office setup.
I know that you're there. I'm just calling to remind you of your sister.
Okay, Apple. And then later on, there's the future office.
Great.
How's your portion of the business plan?
You'll have it by the end of the day.
That's another Apple video. 1985, I think.
I need to watch this again.
So, well, and this continues, of course, like further and further, like, oh, there's some nice VR stuff. This is, I think, Sun's video. Future wedding. And future ecosystem, teaching, future school.
Exactly when and where it's needed. Not all that should.
And what else do we have? We have future. Oh yeah, this is good too.
Yeah.
Future workspace.
Here's the uphill shot of the chart.
This is Sun Starfire.
Car's a bit far away. But I can take care of that with an auto. That's your part. Now, I'll need an ending shot.
Future compositing.
Will you link the camera to me?
Sure.
So, future office presentation, and so on. So, another video that I wanted to share is Fahrenheit. Fahrenheit 4. I think this is from the 50s. So, in 60 something. Oh yeah. Okay, it says there. All right. Awesome. Great. So that's like 20 years earlier. All right. All these things. What? There's one scene that I find particularly interesting here. Oh yeah. Okay. This is kind of similar. Flat screen display. Have you seen? Who has seen this movie?
I have.
Oh, okay. So the interesting bit about this one is though that you can see like something in the like relationship between you know, in this couple is like really like wrong and kind of like, you know, like very different than like the corporate future visions that are all about like very kind of like this ideal environment. And this way makes it to me like almost more futuristic and more real, because it's something that's it's more real social situation. And okay, then I'll jump quickly to of course, wait, let's see, like the classic, of course, Blade Runner. And actually, the model from the opening scene you can actually see at the Museum of Moving Image in Queens. Where is it? Must be right here. Oh, this one. Okay, there we are. the scenes in here, what's his name? Who did the props, like for Blade Runner? What's his name? Who designed the props? Very Yeah, there's one guy who did the props. I can't remember.
Is there no way to sense?
No, it's like he did the product design for the props. I'll remember this. I just forgot it right now.
Okay.
Okay. That's one film I wanted to quickly show. And then there is Primer, which is kind of close to what we're doing, but with time traveling. So that's like a $5,000 like movie that actually Silicon Valley engineers like get together in a garage and invent like a time machine and try to make money with it. So the interesting bit is on how they actually sort of make this like very plausible that this is gonna work, and how the aesthetics of like how they filmed this struck my interest. And these are like just a few like films that I wanted to show. So I want to talk about prototyping futures and possible ways of like doing this by like faking things real. The talk that I prepared is basically talking about nano futures. So there's an experiment. I looked into natural interfaces and how organic interfaces. So sort of like looking into the future of organic computing and how, for example, synthetic biology, if we take it to like human-computer interfaces, how could a future interfaces look like, and how could their behaviors change, and how would we interact with them in the future if we think of like, devices, electronics, and products made of organic circuits, and what would they, how would they work, and how would we interact with them if they would like be organic, and if they would, imagine this messy future of being able to grow devices and like keep them in your home, and like and how would the interactions look like with future products that are not manufactured in Shenzhen and not made of like PCBs. So I actually looked at the behaviors of like food, and this is like a molding orange, and it's like talking to me like by the scent. It's talking to you by like changing the texture. It's talking to you by basically disintegrating. And then this is some shots from just the market where I've been looking at like just basically food and like thinking about food as an interface, and like looking at like food, not food in terms of like eating it and about the quality of like the pleasures of like cooking it, but really thinking about food as material and how would you interact with biological material if it would be data, and thinking in a way that how could we design interfaces that are maybe looking like this, and how would data look like? How could we create interactions that basically allow us to interact with? So this is an experiment that I did. I was looking into like linseed and I took some samples. So this is the back of like a mushroom, and like what would you think? How I could imagine, for example, this could be file browsing, or like how could you create these metaphors for like using like these natural interfaces? So this is another shot of linseed. So in order to prototype like some future where you would imagine, okay, each seed could contain like DNA, like, you know, all the information for the device, and would like self-assemble all these ideas of nanotechnology. What would happen? What kind of behaviors could emerge if devices would actually have these qualities, and like each single piece would contain the information of the whole system? So I started looking into chocolate. Chocolate is melting at exactly at body temperature, and I try to like create interactions with chocolate. This is like chocolate close-up microscopic view. What you see here is like the caramelized sugar and the chocolate butter, and why chocolate is that interesting to interact with is because it melts at body temperature and releases like the sugar with the buttery taste of the fat that melts like on your tongue when it hits the tongue. So, and then I try to create like, this is agar from Japanese supermarket, basically, and I try to like, you know, play around with that material. Then I try to play around with like wood, and you know, how would you have, like, if you sand off wood, for example, what that means if you like interact with data, and then just a lot of experiments. This is magnets, different quality of magnets, reassembling them, and then this is time-lapse animation. That's suggesting like new interactions without actually like exactly saying what it is, but it's like very suggestive video that makes people imagine like what could potentially happen in the future without actually building a device or like making a working prototype. But basically playing with the behaviors. And setting the whole theme like in something that's very common, and people will know what these interactions actually mean. But you know, setting it far in the future, like to talk about like what could smart dust, like make people basically imagine what would be like in a world of like organic smart dust, and how could the future office look like?
So do you think that future artists will use that film at an event in the future like ours?
I think like the discussion about how you talk about the future and how you invent the future is also like, there's the scientific dialogue of like really trying to you know, like you demo or die, and you make objects work, but I also think like you can create these scenarios and fictions that are really fake but can become real by suggesting them. It's just like this is like it's a fine line, like especially if you want to talk like further future out and make people imagine something that you work towards. Yeah, there's more material I can share. This is like something I just put together for the. Cool. All right.
All right. So really quick, I think that Chris's presentation is interesting because that sort of video that you just showed us is something that's eminently doable in a weekend of thing. And I think it's valuable too. It's not just. I don't think it matters to prototype something that's physical or that actually achieves its goal in reality. I think it would be just as good if a group wanted to invent something that was fictional and depict it as being real in a video or in a newspaper or on a website or whatever. So both are like equally good outcomes. So that said, how many of you guys are thinking about sitting here for the weekend to purchase something? 1, 2 people? 3 people? 4? I'm not sure if it makes sense then to brainstorm and take up the rest of everyone else's time who's here for the talks. So maybe we'll do the brainstorming session tomorrow morning with the other people who come to the workshop. And now I think we've definitely got some more food and drinks. And so I suggest we just chill out and talk and informally get to know each other and hang out. You got it? That sounds good to you, Chris?
Yes, yeah. Do you want to do the thing on Sunday? Okay, great.
Okay. So if you are sticking around, we will see you tomorrow. When does this go? 10 and 5. So we'll see you tomorrow. We're going to start tomorrow at 10. If you're leaving now, come back then if you want to participate. Otherwise, let's just chill out. Okay? All right.